In this video, we are going to continue where we left off, and we ended up with discussing mitosis. So again, these are the homologous chromosomes from in your cell. So chromosome 1 from your father and chromosome 1 from your mother. And again, they undergo replication, and what happens is chromosome 1 from your father develops a sister chromatid, and that sister chromatid here is an exact copy of that one. And then at the same time, your mother's chromosome also develops a sister chromatid. So this chromatid here is the exact copy of this one. Then after that, they kind of align and then they divide. And so you ended up with this two daughter cell. But I want you to take note of this. This is the parent cell here, right? This is daughter cell number 1 and daughter cell number 2. Now, notice that the parent cell consists of a homologous pair. So it's a pair, right? So because it's a pair, we kind of describe this as a diploid. Di means two, so diploid, it's a pair of chromosomes. Now again, in the human cells, this would be 23 pairs. But again, so that it's easier to draw, we just have one pair. And then if you look at the daughter cell, guess what? The daughter cell also has a pair of chromosomes and also a pair of chromosomes. So this is also called diploid. So in mitosis, therefore, you have one parent cell, which is diploid, meaning consists of homologous pairs of chromosomes. And then the daughter cells that it produces are also diploids. And notice, this chromosome here is exactly that chromosome here. This is also that. That is identical to this, and that is identical to this. So therefore, you basically ended up with two new cells, but those two new cells are just the exact copy of the first parent cell. Okay? So take note. Diploid parent, two daughters, both are diploids, both are identical to the parent and to each other. Okay, so that's important. So now we go into meiosis, right? So in meiosis, the process is kind of similar, but there are certain noticeable differences that makes it produce different results. So it's still the same. You still have uh, the cells. It still starts with a diploid cell, and then it undergoes replication, so that chromosome develops a sister chromatid, and that chromosome develops a sister chromatid, just like this one. But there is an extra step, and that extra step is after they have been replicated like this, so you now have sister chromatids. These chromatids, these chromosomes kind of decide to exchange materials. What does it mean, exchange materials? I mean this one. So for example, chromosome 1 from father, like this, could cut off one end of its, or one segment of its chromatid, and it could also happen to the other chromosome from mother. But the thing is, they exchange. So this blue missing part from father's chromosome will go to the red chromosome from mother's chromosome. And this missing red chromosome from mother goes to this one. So notice they exchanged materials. Okay, they exchange materials. And this is what we call crossing over. So they cross. And by the way, at first you might say, wouldn't that be dangerous, sir? If they exchange materials, the genetic instruction will be exchanged. So again, remember what our analogy is for the homologous pair. These are equivalent recipe books. So if this is recipe book for desserts, this is also recipe book for desserts. If page 20 to page 40 is about making a leche plan, page 20 to page 40 here is also about making leche plan. The only difference is maybe father's leche plan is a little bit different formulation. It uses more eggs. Maybe mother's leche plan uses more milk, right? So there's subtle differences there. But the point is, if they exchange, for example, page 20 to page 40 here is exchange for the page 20 and page to page 60 of this book, guess what? This would still have a recipe for leche plan. This would still have a recipe for leche plan because they exchange the exact pages. So it doesn't affect the function. Okay? So that's key there. But they never exchange like separate segments, because if they exchange separate segments, then that would be different, because for example, if you have a segment here that causes the proteins for your eyes, it could not be exchanged for a segment that encodes for the proteins for your skin. It might have a different effect, right? So it's only same segments that are exchanged. So again, this is just kind of represented by the tips of this chromatid here and the tips of that chromatid there. Okay, and then what happens is, remember in mitosis they kind of like this, they align. In mitosis they align like this, and then they get separated. This does not happen. Instead, what happens is they will align, but in homologous pairs. What do you mean by in homologous pairs? Remember, meaning chromosome 1 from father will align with chromosome 1 from mother, and chromosome 2 from mother will align with chromosome 2 from father, and so on up to chromosome number 23. But again, here, because in our example we only are drawing one chromosome, that means this and that will just align, but they will not align vertically. They will align horizontally, meaning this will kind of be like that, and this will be like that, and then obviously that is where the red tip is. This is the blue tip. So instead of aligning kind of horizontally, vertically, they align horizontally but in pairs. One, one F and uh, one F and one M. One from your father, one from your mother. But well, technically speaking, it's not really from your father anymore, because you have exchanged some material. So we actually call this recombinant chromosomes, because they recombine certain parts. But that's beside the point. The point is they kind of align, but instead of vertically, just like in mitosis, where one is up and the other is down, this one they align horizontally, and this goes for chromosome 1, chromosome 2, chromosome 3, up to 23. But again, in an example, we only have one. And then what happens is this divides. So when this divides, that means this goes to here, right? And then this one, this goes to here. Right? Makes sense? So now, you now have this one. And then that's the end of meiosis 1. Okay, in meiosis 1, the difference is before they align at the center, they kind of exchange some materials. We call this crossing over. But again, the key there is they only exchange certain segments. If we use an analogy of books, they exchange exactly the same pages or the same genes, the same segments, so that it won't be doing any bad effects to the organism. Then after that, they align. But unlike in mitosis, where they align in a position of top bottom, so chromosome 1, chromosome 2, chromosome 3, up to chromosome 23 in one vertical line. In this case, they align in horizontal line, wherein you have chromosome 1 from chromosome 1s, chromosome 2s, chromosome 3s, 4s, 5s, 6, up until 23. Then they divide at the middle, so therefore you have a cell here, but the cell is still made up of chromatids, and also here it's still made up of chromatids. That's meiosis 1. In meiosis 2, this further divides. Okay, so this becomes this cell, this becomes that cell, and then this one maybe becomes that cell, and this one becomes that cell. So let me fix my arrow. So what does this mean? That means this red with a blue tip will kind of go here. Red with a blue tip. While this completely red one will go here. And then this blue with a red tip kind of goes here. While this completely blue one goes here. Okay? And that's it. That's the end of meiosis 2. So this is now the process of meiosis. Now, let us examine what's the difference in terms of the results. Well, you still have a parent. That parent is still a diploid. Right? Remember, diploid means a pair. Homologous pair. Again, in human beings, it might be 23. In our example, it's just 1. what is the end result? Notice that this is the end result, 4 daughter cells. And these 4 daughter cells is, I call this daughter cell 1, daughter cell 2, daughter cell 3, daughter cell 4. Notice, are the chromosomes in their pair a pair? No, they are not made up of pairs. It's just half a pair. So therefore, this is what we call haploid. So in meiosis, what is produced are haploids. Meaning it's not a pair of homologous chromosome, it's just one chromosome, right? One chromosome. And then you might ask, why is that? Well, because again, as you learned from grade 8, meiosis is for the production of sperm cells and egg cells, so gametes. And these gametes, remember, they have to combine. Remember during our discussion in the first reading about some parts of reproductive system, it's actually an egg cell and a sperm cell that kind of joins together and forms a zygote. So that means half of the DNA material is coming from the sperm and half is coming from the egg cell. So if a haploid sperm cell meets another haploid egg cell, they will become a diploid when they join together. So that's the idea. So in mitosis, you end up with diploids, but in meiosis, you have haploids. But more important, notice that these cells are different. This is completely red. This is red with a blue tip. This is completely blue. And then this is blue with a red tip. What does that mean? That means this is not the same as that, is not the same as this, is not the same as that. So the 4 haploid cells are kind of different from each other. And if you're wondering, why is that important, sir? Well, that's important because think about it. If sperm cells that are produced in meiosis would be identical, just like in mitosis, remember in mitosis, the cells are identical. That means that all the sperm cells coming from your father will be identical. And all the egg cells coming from your mother will be identical. So if they're identical, then if they produce an offspring today and they produce an offspring 5 years from now or 10 years from now, guess what? Those offspring will be identical twins because they have the same genetic material, right? But you know that does not happen. You know that your siblings, you know your kuya is different from your ate and different from your manghud because the sperm cells are not the same. In the same way, the egg cells produced by your mom, because it still undergoes meiosis, are different. So they are different, but at the same time, yes, they're different, but they're still based from this. They are still based from this, from mother's chromosome and father's chromosome. It's just that they cross over, and because of the crossover, instead of pure blue and pure red, you now end up with pure blue, pure red, red with a blue tip, and blue with a red tip. That's exactly the reason why they cross over. Because if they did not cross over, then this would be blue, blue, red, red. This would be blue, blue, red, red. This would be blue, 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 red, red. This would be blue, blue. This would be red, red. So they will be identical, or well, not identical and blue, red, but still, it's not as variant as when you have four different cells. And if you have 4 different cells, that means that's why your kuya is kind of different from your ate, even if you came from the same person, gave the same that your sperm cell came from the same father and your egg cells came from the same mother, because each egg cell and each sperm cell are kind of different each time, right? So you have this to be thankful why your siblings are a little bit similar to you, because you are based from the same chromosomes, but at the same time they are a little bit different from you because of this crossing over. Right now, think about again, if they did not cross over, then sperm cells would be identical, egg cells would be identical, and therefore you would be there would be less variability in your family. And also And also take note that when this cells kind of interchange materials, here I'm just simplifying it. In this case, they are interchanging the tips, but in the next batches of meiosis, it might not be the tips that they exchange. It might be some other pages. So meaning, even if you have these four kinds, it doesn't mean that you only have these four kinds, because remember, the number of the pages that they exchange, the segments that they exchange, could also change from time to time, and so you could have different uh, uh, configurations of the genes. But definitely, you are still similar because you are based from the same genes. It's just that there's this crossing over, and therefore it includes variability among siblings. Okay, so that's the idea. But again, before you divide, you have to undergo cellular replication. This single chromosome structure must develop a sister chromatid so that when you divide, you would have, when you divide and then divide again, you would have haploids. So meiosis creates haploids, and mitosis creates diploids. So in fact, mitosis creates two daughter cells that are completely identical to the parent cell and completely identical to each other. Meiosis Meiosis, even if they're based from one set of homologous chromosomes, because of the crossing over and because of two divisions, meiosis 1 and meiosis 2, you end up with four cells that are first haploid, not diploid, and you end up with this daughter cell is different from this, is different from that, is different from that, increasing the variability of genes that can be created in your family.